Cell cycle. So for many of you, we've all seen to some degree cell cycle, some more than others. So you've had introductory cell cycle. The thing, originally when I first started teaching this course, I wasn't going to have cell cycle in. I was going to take it right out. What a lot of cell biologists do, they talk about mitosis, and they talk about it as almost like an isolated event. And what a lot of people do, they memorize mitosis, but they don't really understand where mitosis fits in the rest of the life of the cell. So we're going to talk about cell cycle today, and give you just, if I be revisiting some of it, like some new concepts as well. And then that will set the stage then to incorporate mitosis and kind of go zoom in one particular stage. But there are multiple stages to cell cycle, and that's what you need to make sure you understand. So as far as cell cycle, it covers a few things. It covers G phases that we're going to talk about today, which very important things happen. S phases, which critical things happen. And then M phases where things divide. You get two cells, they divide, and then the whole process starts again. So when you're going through this, make sure you understand that sequence of events, because again, this lecture will assume that you do know that, and that's what I'm going to go through. So what do you need to know? Obviously you need to know the stages of the cell cycle, right? How it feeds into mitosis. Who's who in the system, what needs to happen before you move to the next stage. Again, very important in terms of that overall picture for cell life history, if you like. The importance of each stage, what happens in each stage, and we're not going to dive into any one of them in great detail because we don't have time. When you do molecular biology, you lecture on genetics, you might dive in a little bit with these, on physiology and development. But with those, we're going to just basically make sure you understand the basics of that. The key proteins that control the cell cycle, the proteins, the quality control proteins are everything in this. That word that I always talk about, temporal, this is probably the most important lecture for temporal, because everything is about timing. If things are not done right and properly in the right time, cancer. Okay? Cancer and cell cycle are pretty much coupled in every single way. Okay? And so there's 1,000 labs on this planet studying cancer, but they're studying different aspects of the cell cycle, to try to understand more about how cancers arise, and secondly, how we can potentially use drugs to target these different stages to try and alleviate cancers or prevent uncontrolled cell division, which again is all relevant to cancerous tumor growth and tumor development. So, cell cycle. Cycle of duplication and cell division. Again, it might seem obvious, but make sure you know it for the exam. So, I'm going to ask you questions at the beginning, assuming you know the basics of cell cycle before we dive into specific mechanisms that we'll touch on for a little bit. There's obviously multiple rounds of cell division. That cell division is obviously critical because once you divide, then you're going to get new cells which need to start checking themselves out and making sure they're good before they divide all over again. And again, that process is really, really fine-tuned.
Can you turn off one of the lights? It's hard to see.
It's hard to see.
Yeah.
What is it? This is for the...
I think there's a light switch back there by this door. I don't know what it is. It says light on there, I think, main light.
I'm used to that. Does it look like a light switch? Yes, so.
Come on, if we close our eyes, we can turn off the lights.
Is that better?
Yes.
Somebody changed it. Okay. So, cell division, the basic concept for a cell cycle. Yeah. Thank you. I don't like these. I love it, but don't tell everybody else that. So, there are multiple concepts associated with cell cycle. You can copy, you can pass that information. The copying of the information and the passaging into the next generation, again, is critical. That's everything. So if there's anything that changes or doesn't happen properly, we have systems in place in the cells that can check them things out and say, wait a minute, don't move on until we've checked the DNA. Let's make sure it replicated properly. Let's make sure the cells grew to a certain size. Let's say when you divided the cells, there's not damage in the DNA. We have all these checkpoints that can allow the cell, to some extent, to check them things and make sure they're good. If they're not, we can hold the cell, and that's something we're going to talk about. But in many cases, when you have mutations arise in proteins that are involved in these checkpoints, that's when the problems arise, because you can't control things that maybe go through that shouldn't go through. Damaged DNA, cells that are not big enough, cells that are wrong shape, all these types of things which will be passed on into the next generation. So make sure that just kind of basic concepts you understand as far as just importances for the cell cycle. How do cells duplicate their content? How do they carry the information? So the duplication is based on what we'll talk about is the S phase. You replicate the DNA within the S phase to allow two cells to ultimately come from that one cell which you originated from. How does this stuff split and then divide? Again, we'll talk about mitosis in a couple of weeks, in a couple of lectures' time. How does a cell coordinate all these steps? And again, this is based on two key sets of proteins. Some kinases, like normal, and some proteins called cyclins that we're going to talk about. Absolutely critical for being able to monitor and make sure that things don't move to the next step in the cell cycle until the cell wants it to. So, basic things, I'm not even going to go into this because this is stuff you should already know, but make sure you review it when you're reading over the lecture because it's the most basic stuff. The duplication, of course, the segregation of the DNA into two cells that have been identified, make sure you understand them two important things. Each cell receives a complete copy of the entire genome. If there are mutations in that mother cell, they're going to be passed into them daughter cells, that progeny. So again, that's why there's systems in place to make sure we can hopefully correct these things before the DNA gets replicated and then gets decided to go into two different cells. So again, quality control is everything. And also, it's not just the DNA. All the macromolecules and organelles within that cell need to duplicate as well. So you have all the DNA, all the stuff in the cell needs to be duplicated. So you have two sets of it. So then that can end up into two cells and be divided, and they can go off and do their thing within the cell cycle. So again, the basic steps, make sure you have them down at the least. The speeds, the timing of these things, of cell cycle, can be very variable. Some organisms have very fast cell cycle phases. Some organisms are a lot slower. And again, it depends on the organism. And obviously over evolution, these systems have evolved to be faster or slow, depending on what type of organism they are, what demand they need, what nutrients they need. For cell cycle to happen, there needs to be various types of nutrients and molecules that are talking to doing all these different biological processes to allow the cell to go through the different phases. Okay? So again, I'm not going to test you on all the numbers, but make sure you understand there are different rates to the cell cycle for different organisms. These two things, like I mentioned before, are key. Make sure you understand that these two things, they're key. Correct sequence of events. Obviously, there's a specific sequence of events that has to happen every time. And then secondly, when they happen, they have to happen at the right time. If they happen too fast or too slow, that can be detrimental to them two cells that are going to ultimately come from that one cell. And again, you have a ripple effect on the rest of the cells. And again, lots of that problems are associated with a whole array of types of cancer. Okay? In terms of the phases, so this diagram, you guys are probably still dreaming about GPCRs. You need to start dreaming about cell cycles. Okay? Not because of just knowing the cell cycle, but what you'll do is you'll know where the mitosis fits in. And as a result of knowing where mitosis fits in the cell cycle, you'll also begin to understand after we do the cancer biology lecture, where cancers can arise. So start integrating the stuff now. Don't do this at the end, where it all goes into your short-term memory, and 2 days later it's gone. Okay? Read it now, and then when the next lecture comes on the cancer and on the mitosis, this stuff will go in, hopefully, to your longer-term memory. That's why we read these things over and over again over a longer period of time. Otherwise they're not. They're just going to come in and leave. You do the exam, it's gone. Okay? So for the cell cycle, You have the end phase that we're going to talk about, which is mitosis and cytokinesis. You have G1. What does the G stand for? What's that? The G stands for gap. Okay? Gap 1, G1, G2, S phase, or G1, S phase, G2, and N phase. There's no variation in that. That's what happens. Okay? So, make sure, if you remember nothing else for this lecture, you understand this. Okay? Because if you understand this, even if something's not there when you first see it in the exam, you may be able to make an educated guess based on remembering these steps. And if you can remember what happens in these steps, then hopefully that can lead you to the answer. So, once you have G1 phase, you will lead into S phase to duplicate or replicate the DNA. Then things will happen to basically get the cell, make sure it's ready to then go into mitosis and do everything else. So, mitosis, cytokinesis, both of them are part of the M phase. Some people think M phase is just mitosis, that's wrong. The M phase is the mitosis and the cytokinesis together. Okay? So this M phase is here. This whole phase here, everything else is called the interphase. So the cell, for most of its period, is in interphase stage somewhere. The M phase isn't that long, very fast. Okay? So even though when you've done your biology courses before this, I can say most of the stuff when you talk about this is about mitosis. But mitosis is only a small portion. It's really, really important, of course, but it's only a small portion in terms of what's happening in a given cycle of the cell. There's lots of other things happening. 90+% is through the G1, S phase, and G2 phase. S phase, make sure you understand this process, very basic process of replication, and we're going to talk about replication at the end before we finish. The S phase is always flanked by these two G phases. The G1 phase has to come, of course, before the S phase, because things need to happen in G1 phase before the phases transition into S phase. I will not start replicating my DNA unless I've made sure my DNA looks good. Okay, and there's systems in place to help in checking that DNA, make sure it's not damaged, make sure the cells are the right size before we start duplicating everything. And if we don't, we hold that off and we can hold that transition, stop it, and so things basically get rectified. Then it will move on to the next stage. And then once you go through S phase, then you go into G2, where things again, there's last few things that we'll briefly talk about where the cell is growing a little bit, making sure it's the right size, things are where they need to be before we start to do mitosis, before we go into prophase. Okay? So that mechanism of S phase and then G2 phase is all preparation for the big game, which is this. To then say, once we have this cell down, then we need to start making two of these and dividing them. Okay? And you won't do that. The cell will try and not do that if any of these ones something's wrong. It has proteins in place, and these two proteins that we're going to talk about are called CDKs and cyclins. Yeah.
Sorry, real quick. Is it so order? It goes G1, S, G2, and N phase.
G1, S, G2, and then N phase. Yeah, always. Yeah. Okay, so we've talked about this. Checkpoint proteins. So this is what we're going to spend most of the time on. So if you do a class, any class, doesn't matter what class it is, if you do, especially do cell cycle or anything associated with it, the majority of the lecture will always be on checkpoint proteins. So that's what's doing 99% of the work. They're either these cyclin-dependent kinases, CDKs, or the cyclins, which actually modulate the CDKs. Okay? So the CDKs are kinases that do things, phosphorylate, obviously, but their ability to phosphorylate and their ability to be synthesized and do the thing is heavily modulated by a second set of proteins, the cyclins. So the cyclins are what really shape how these CDKs do various things. And what the things they do are act as these checkpoint proteins at different stages of this cell cycle. We're going to break them down a little bit today. So as far as the different, make sure just before we start the CDKs, make sure this slide is clear before you start reading through CDKs when you're going over the lecture. The sequence of events, the big sequence of events, stage after stage after stage. And then when you start to read the CDKs and the cyclins, you can see where they all fit. If this doesn't make sense, the CDKs and cyclins are going to mean nothing. So make sure you spend some time. If it's already there, great. If it's not, extra couple of bits of time on this, and then shift into the CDKs and cyclists. So, as far as 4 things I could ask you, if I am going to ask you in the exam, okay? So as far as things that the cell cycle is important for in terms of control, ensure proper regulation of DNA replication is one thing obviously that's critical. Duplication of organelles, so you have 2 sets of them. Growth of the cells need to grow to the right size before they start to do the dividing, going through M phase. And obviously finally the cells need to divide. So again, procedurally, in terms of how things happen, make sure you have this down for the exam. And each one of these, again, is temporally controlled to the maximum. Because if you don't, really big problems can arise as far as disruptions, alterations in the way these cells work. Okay. Regulatory protein networks. When we talk about regulatory protein networks, we're talking about the CDKs and we're talking about the cyclists. They regulate everything. The timing especially, when you can shift between this stage, this stage, this stage, and this stage. Okay? And they will keep you there or push you through it, depending on what time it is, when you need to do that. Okay. Learn this slide. I put this in because this is a summary of the last 10 slides that we've just talked about. So make sure this slide makes complete sense as far as the strategy the cell takes, the trajectory the cell takes from the point of having new cells all the way through to dividing all over again. Okay? And so when you look at these, the basic things you should be able to see is, at least for the M phase, when you produce them new cells, in this phase, this G1 phase, the DNA and various other things are looked at to make sure everything is good. The DNA is not hopefully damaged, and if it is, there are DNA damage mechanisms, repair mechanisms that we're going to talk about briefly that will help out in this situation. And you can end up have mutations that arise in these pathways that, again, if you break the DNA repair mechanisms, you don't repair the DNA, and you pass on cells that you don't want to pass on with damaged DNA, and that's where you get mutations in very key proteins that are involved in cell division, allowing the cells to be uncontrollably divided, and that's tumours. Okay, so again, lots of work is done, it's being done for a long time. The proteins associated with G1 and how they correlate with various types of cancer, etiologies of cancer. Checkpoints. So as far as this, so coordinates machinery. So the machinery, the CDKs, the cyclins that we're going to talk about. Again, these are the 3 things you need to know besides the 4 things that we just mentioned before. So, just reiterating, the cell division, the DNA replication, and the segregation of the chromosomes. We will do all this in mitosis, and we'll go a lot deeper in this. So if this is still hazy or you can't remember this from previous courses, we will go into this. You guys will be all set. But again, it's the checkpoint proteins that do all this stuff. And the proteins come in 2 forms, at least 2, kinases and phosphatases, and then obviously the cyclins that we're going to talk about. And so that means, just like many of these mechanisms, it's a series of phosphorylation and dephosphorylation mechanisms that are associated with switching on or switching off cyclins, or switching on or switching off the CDKs, which are modulated by the cyclins. Okay? So the CDKs do a lot of this stuff in terms of pushing the cell through one phase to another phase. But the way they do it, the rate they do it, the timing they do it, is based on a lot of it based on the cyclins that are controlled. And so that whole process is based on the cyclins being broken down and switched on very dynamically. And again, very in terms of temporal control is critical. You need to be able to get rid of these things at certain steps, and then you need to make these things at certain steps. And so that ability to break them down and build them up is critical, because if you don't do that properly, the cell will not know where to go into the next phase, or it will go into the phase too quick. So again, it's all about timing of these CDKs, these cyclins that are associated based on the phosphorylation and dephosphorylation mechanisms. So just for one example, is in the G1 phase, when the cell wants to go into S phase, where the cell is going to replicate the DNA, there are proteins that need to increase their levels of transcription and translation. They need to be switched on. So they can push the cell from G1 phase into S phase and initiate what's called DNA replication. If them proteins, them cyclins, them CDKs don't increase late in G1, then it won't be able to transition into S phase. So it's not that these phases just happen and then they go up. For the cell to go from one phase, from a G1 phase to an S phase, these proteins need to start being transcribed and translated early, or well before the S phase. So at the end of the G1, and I'll show you a graph in a minute, where it shows basically the dynamics of synthesis and breakdown of these CDKs and cyclins. Because that's what that temporal control is what allows you again to shift between phases and allows you to do it properly. Same thing for mechanisms associated from G2 to M phase. For me to go from G2 to M phase, there are cyclins and CDKs that need to be switched off. There are cyclins and CDKs that need to be switched on. And that will allow me to push myself through into M phase at the right time. So again, the timing of breakdown and build-up is critical for this. Okay, so cyclins allow kinases to regulate. Modulate is a good word. Okay, modulate is a word that basically means it's a word you'll probably never ever hear except in biology in general. It's when it shapes something else, it fine-tunes something else. So the CDKs want to do something, but they're regulated, fine-tuned, modulated by these cyclins. And so these cyclin-dependent protein kinases, their expression, just like the cyclins, is what's called cyclic. It oscillates up and down, depending on the stage, depending on if you're at the beginning of the stage, beginning of S, late S, beginning of G1, late G1, wherever it is, there will be activation and switching on of genes that are involved in producing them CDKs or cyclins at the right time. So then again, you can push the cell into the next stage. And in general, CDKs, for most of these diagrams, especially in the book, in the Essential Cell Biology book, they're in general big red proteins. And then these cyclins come along and basically bind to the CDKs. And one of the mechanisms we're going to talk about for this kind of modulation today is the opposite to what we talked about for many of the phosphorylation mechanisms. Most of the ones we talked about previously were where phosphorylation causes the protein to become activated. In this case, dephosphorylation activates these types of proteins in some cases. So again, that's why there's a multiple effects by phosphorylation and dephosphorylation. It's not just phosphorylation activates, phosphorylation can also inhibit it. Okay? But it's all about interaction again between these cyclins and CDKs, and it's about these cyclical oscillating CDKs and cyclins that will give different steps that can allow these pathways to shift from one phase to the other. So once these things are switched on, they will then initiate or help drive one stage to another stage. And so, for example, there's a nice graph here, a nice picture where this is showing the concentration of one of these cyclins. Okay? And so we know this M cyclin is a cyclin that, when it's switched on, it's going to help push the cell through mitosis, or into mitosis, through mitosis. So if you looked at its expression levels over time, you would expect its levels to be low during interphase, during S, G1, G2. You don't need it. And then suddenly you need to switch it on in the M phase, and then you suddenly get a big increase. And you don't just get an abrupt increase right here at the beginning of mitosis. Remember, these things start to switch on, if you can imagine, almost in the last 30% of the stage before. You start to get transcription, you start to get translation of these proteins, so cyclin will go up. And it's gradually going up to the point where it's nearly at a maximum, at the point where at the end phase, where mitosis begins. And it will stay high because during mitosis you need this M cyclin. It does a few different things that we'll talk about in mitosis. So it's involved in driving this G2 into M phase, and it's also involved in M phase doing a few things, like I said, we'll talk about when we do mitosis. And this is the type of thing you'll see with any of the cyclins and CDKs, is that there's obviously a correlation between the specific cyclin that regulates a specific CDK and its levels of transcription and translation or breakdown dependent on what stage you're at, whether it's G1, S, G2, or M. So again, we have to control these with really, really high accuracy, otherwise, again, there's a lot of problems for the cell. And then G1 is another example, again, to drive G1 into S phase to prepare the cell to say we're going to replicate the DNA now. We've checked the DNA in G1, everything is good. We now want to replicate it again. There are proteins, there are CDKs and cyclins that need to start going up during the end of G1 that are going to help push, if you can imagine this was S phase in this case, let's say, and this was G1. Some of these, what are called S-CDKs, because they're involved in the S phase, they will start to increase here. And they'll go up to the point where they're increasing very high as they go into an S phase. So them CDKs that are specific for driving into S phase need to increase just before S phase begins, just like the M-cyclin does just before the mitosis. And by doing that, it's not just doing it randomly, it's doing it because it's going to initiate a bunch of mechanisms. And for this S phase, for these cyclins to increase, one of the things it does is it modulates specific S-CDKs that would normally, one, help them push into S phase, and two, prepare the DNA for DNA replication. There's a whole mechanism for regulating for DNA replication. And just like for transcription, you have to have a whole transcription initiation complex that happens in eukaryotes. It's a similar thing for DNA replication. You need to get a load of proteins that come onto a big site that's very conserved to produce this complex that will allow drive replication from them specific places. And we'll talk about the mechanism of that later. So we're not going to talk a lot about the mechanisms of these different ones. We'll talk about some key proteins. But one mechanism we'll talk about a little bit more detail will be the S phase. So this is an example of one of the, if you look at, there's actually two of them here. So if you look at the SCDK, so the SCDK, as I mentioned, it goes up here. You see how it starts to increase here. And then it's priming this part of this cell here so it can go through S phase. It does that because this CDK is activated by this S-cyclin. The cyclin comes in and binds to it and allows it to start doing its thing, which is to start promoting S phase. And a similar scenario for the M phase. The M phase M-CDK that's involved in mitosis, pushing the cell from G2 into M, and then doing things in M as well, is only going to happen because the M-cyclin transcription translation goes up. It then grabs hold of the CDK and says you can start doing your thing. So depending on what stage we're talking about, we have specific CDKs that are activated or regulated, modulated by specific cyclins. And again, their expression and their breakdown is cyclical. It goes up and down very fast, depending on the stage that we're talking about. Phosphorylation. So as far as the phosphorylation, I mentioned before about the phosphorylation, dephosphorylation, in terms of the directionality of how that affects the protein complex.
To verify the lines on the picture, that's just like the amount of.
That's just the amount of it. Sorry, this is like the previous diagram. I'm just showing like where the levels are at. And the reason why we have this in as an addition to having this is because we want to, in the book especially, we want to stress the fact that these things need to be transcribed and translated at a perfect time. They need to be broken down at a perfect time. So you can literally follow the functionality of that stuff based on this level. And so it's very low.
Just present at those points, and because of the increase.
When you need to push through from whatever S to G2 or G1, you now need to start transcribing, translating that CDK and the cyclin that's involved in it. Then the levels will go up, then it will start to do its thing. And they'll stay high until you don't need them anymore. And when you don't need them anymore, then you'll go through a ubiquitination pathway, a proteasome pathway, and you'll break down the proteins. Okay? Again, a very quick process, but as far as control is critical for any one of them steps to exist properly. So transcription, translation of the cyclin genes to produce them. But again, the thing that we've already talked about, but is just as important, if not more important than we talked about before, is the degradation. Once you've switched them things on, they've done their job, and then you need to, let's say, leave M phase and go into G1. You need to start promoting G1 phase proteins, but you need to get rid of the mitosis protein, the mitosis promoting proteins. You don't need them anymore. So you need to, at any one of these stages, when this thing goes up, this part here where they go down, this is critical because you need to break them down really fast and then start translating ones that you need very fast to start pushing the cell into the next phase. So there's a constant oscillation between these, and that allows them to be able to divide very quick, and hopefully do it properly. And that's based on the proteasome pathway that we've already talked about. The ubiquitination that we've already talked about, the proteasome pathway. I'm going to ask you a question again on that. So make sure you say, oh, we've already done it. Well, make sure you have that down for the exam, because this is important for breaking down these cyclin protein CDKs very fast. Okay. One specific mechanism I want you to know for the exam as well, and it's a very cool one. So, in terms of regulating these cyclins, I just mentioned about the ubiquitins and the proteasomes. These types of mechanisms, make sure not just that you know the kind of A goes to B goes to C, but make sure you can kind of visualize it in terms of, you're not going to have to draw it, but if you can visualize this stuff, again, it will make more sense. And then I say to everybody, if you can't draw this stuff, you don't understand it. Okay? So draw these pathways out, and if you can, you're all set. There's a protein, an active cyclin CDK complex that are doing whatever they're doing. When you don't need it, you get these ubiquitin chains that get added. Now in this case, there's a really important protein called APC, anaphase promoting complex. This APC is critical for this process of adding this ubiquitin chain. So when I don't want cyclin or CDK around anymore and I want it to go down, I basically push it through this pathway and I activate the APC. The APC is one of the proteins involved in, one of the complexes involved in ubiquitination. And once that chain gets added to this cyclin, remember this is the CDK, this is the cyclin, it will cause that cyclin to come off and get broken down. Proteasome dependent breaking down of the protein. CDK stops working, because remember the CDKs need to be activated by these cyclin-dependent mechanisms. So the ability to break down and build up the cyclins very fast is what allows these CDKs to be able to be switched on and switched off for a different period of time. Yeah.
So cyclin is dependent, there's varying cyclins
Correct.
depending on the cyclin phase that we're in, but are there varying CDKs that match with specific cyclin?
Yes, we'll talk about that. So, types of CDKs, cyclins that exist. G1 CDKs, got G1S CDKs, so CDKs that are involved in putting the cell into G1, ones that are involved in transitioning between G1 and S, ones that are involved in replication, the S phase, and then ones that are involved in mitosis, cytokinesis, pushing the cell into that phase and keeping it there and doing things in there. Okay? And there are specific cyclins associated with these different CDKs. And then things that you don't need to know this for an exam, and we could have 5 more lectures on this particular bit, but there are a bunch of CDKs that are associated with these CDKs and cyclins, based on the mechanism being a lot more elaborate than what we're talking about right now. But the point is, people have identified various other proteins that interact with these CDK pathways. Not just the cyclins, but a bunch of these other CDK, cyclin-dependent kinases, that actually are associated with these pathways. In G1, in S, in M phase, G2 phase. Okay? And again, time-wise, for time reasons, we don't have time to go through it. But again, the take-home message that I want you to get is cyclins do help make sure the CDKs do what they do at different stages in this pathway. Okay? Okay, so this is what I was talking about. Right, so as far as the M cyclin, this M cyclin here. So this is where you want...
Since the CDKs have to be bound by a cyclin for them to be active, do they have to undergo degradation too?
The type of CDKs? Yeah, some of the CDKs can hang around and they can be modulated by cyclins that come on and off. But in most cases, the CDKs can just get broken down as well. Because again, you don't want kinases around when they're not doing it. Remember, you have specific, if there were one CDK that was going through all the pathways, and depending on who talks to it, it will do a different function, then you can keep it there forever. But based on having different CDKs at different times, you need to break down the CDKs and cyclins. But the thing that stops the CDK from doing anything in the first place is to get rid of the cyclin. Okay. Yeah.
So the APC only ubiquitinates the cyclin or the whole complex?
It ubiquitinates the cyclin gets degraded, and once the degradation happens, the CDK is useless to that, basically. And then you have ubiquitination pathways that are proteasome-dependent pathways that are associated with degradation of the CDK when you need to get rid of it. So in most cases, unless I specify, proteins are always going through some proteasome ubiquitination-dependent pathway. So there might be a sequence of events of who needs to be broken down first. And seeing as the cyclin controls the CDK in most cases, then it's quickest, more cost efficient to get rid of the cyclin, and then you can do what you want with the CDK after that. But again, remember, there are specific CDKs for specific stages within the cell cycle. So this is the only one that we're going to talk about the mechanism, if you like, about how these cyclins can be activated or deactivated. And the reason why I chose this one is because this is counterintuitive to, again, some of the way, a lot of way your mind might be right now about how kinases do things to complexes versus phosphatases. So when you want the M cyclin to stop, the M CDK to go up to start doing things, you need first the M cyclin to come in. And the M cyclin will bind to the M CDK. And then you can start to drive mitosis, hopefully. The reason why this is not allowed to do this until it's the right time to do it is because this complex is inhibited by a protein. So this inactive MCDK here will be, the cyclin will come in, it will basically come in, the cyclin will come in and bind to this CDK. And then, what the problem is, there's an inhibitory kinase. Remember, this is where it's kind of counterintuitive. There's an inhibitory kinase that when it phosphorylates the red one, so it phosphorylates this CDK, it keeps it inactive. Okay? So as long as it's phosphorylated, it's not doing anything. And then a phosphatase comes in, CDC25, and this removes the phosphate from this complex. By removing the phosphate, you now activate the CDK. So in this case, the phosphatase is the activator, and the kinase is the inhibitor. Does that make sense to everybody? Make sure you know these two names. So for the kinase, it's inhibitory, WEE1, and for the phosphatase, CDC25. Absolutely critical for controlling the levels of this activity of the M cyclin MCDK pathway, which is critical for driving into mitosis and doing things that we'll talk about in mitosis. So these two proteins are right there, and it's their levels, their ability to do what they're doing that's going to fine-tune this pathway. So when we activate, we want M cyclin MCDK to do something, we need to get rid of this, or stop this guy from working. And the way we do that is by bringing a phosphatase that brings off the phosphate. And now myself and my M cyclin can go and do what we need to do. Okay? So mechanistically, this is something that's critical for M phase to even exist. So make sure you know this diagram very well for the exam. Okay?
Is that a similar trend for all the cycles?
No.
Just how?
There's examples of both. There's definitely examples of CDKs being activated by phosphorylation. But this is one example where it does the other, and that's why I like to do it. Okay, so as far as I put this in again, it's kind of a summary, it's just kind of a reminder of a few things, but this again, I will ask you these I will expect you to know these for the exam, for the questions. Okay, so the cell cycle control system can pause the cycle in various ways. So instead of just pushing me forward and keep going, I can also obviously hold the system. The checkpoint proteins are critical, pivotal in that ability to hold the system until you need to carry on through to the next phase. The phosphorylation states of them proteins is everything, well, in terms of allowing them to become active or inactive, which goes hand in hand with the checkpoints. The checkpoints are the proteins that need to be phosphorylated and dephosphorylated. So their state of phosphorylation is critical for the timing of these steps. The types of proteins that are recruited, depending on the stage, you will have different CDKs, you'll have different cyclins, and you'll have a bunch of other different proteins that have different stages that we're not going to have time to talk about. But at least for what we have talked about, make sure you understand that. And the speed is critical. You have to be able to build these cyclins and CDKs up, especially the cyclins, up and down, and get rid of them and bring them back and forward very fast. And if you can't do that, that's when the cell will hold in a position until something starts to be fixed, and then you can move on. And again, you can have a series of mutations in these cyclins or in these CDKs And mutations in these CDKs or cyclins can result in these stages moving on through to their next stages without checking things, or not letting the stage happen at all. And so you'll have cells that will just die, they'll just go through apoptosis because they can't move to the next stage. Or you'll have cells that are produced and then divided that have not been checked out before they've gone through that whole process. So again, mutations, this is a huge component as far as mutations that arise over generations, where you have cancer cells that proliferate and you produce mutated cells over and over multiple generations. And a lot of it feeds into these problems associated with the cell cycle. In terms of pausing, so these three statements you should know, okay, for the exam. At the G1 to S transition, it uses CDK inhibitors to keep the cells from entering S phase and replicating their DNA. What does that mean? It means there are proteins that can say, before you activate SCDK and go into S phase, we need to keep SCDK inhibited. We need to stop these proteins. And then when it's time to be activated, we can remove that inhibition. And then the SCDKs and the other proteins can then start to prime the cell for S phase. So, but we can hold them in that position with various inhibitory mechanisms until it's time to move on. At the G2 to M transition, there's a suppression of M-cyclin. We just talked about what's the suppression, how's an M-cyclin stopped until you need it to work in mitosis. Good. Phosphorylation, what's the protein called? WEE1. WEE1 is critical for this whole concept, for stopping things until it's time to move on. And then the phosphatase, which is called what? What's the phosphatase called? 25. CDC25 then comes into play, and that will remove that phosphate, and now M-cyclins, MCDK, you will start pushing the process forward. And then delaying the exit from mitosis by inhibiting the action of APC, thus preventing the degradation of M-cyclin. So when M-cyclin is finished, you then need to switch off M-cyclin. Right? So M-cyclin's here, then it goes high, you go through mitosis, cytokinesis, then you need to switch it off. Now I'm in G1. M-cyclin needs to be switched off. And then a protein complex called APC will switch on to check the DNA and be involved in, it's involved in DNA, it's involved in regulating the phosphorylation state of various proteins. APC is really important in checking things out after, post-M phase. Yeah.
For number one, the CDK inhibitor, does that just keep cyclins from binding? At the G1.
So there's multiple CDK inhibitors. There's ones that inhibit cyclin from binding to CDK. There's multiple ones that don't touch cyclins, but they stop CDK from having their effect on the protein. It depends on the inhibitor. Some of these inhibitors directly bind to cyclins, or want to bind to cyclins. Some of these inhibitors actually target the CDKs. And so if you're in this phase here, what this SCDK is involved in is promoting S phase by it's involved in the priming process to generate a pre-replication complex associated with DNA replication. So that DNA replication complex is going to need to be there to start generating DNA in S phase. It's not going to do that until these inhibitor proteins come away and the cyclin and CDKs can start helping promote that. And specifically the SCDK. Okay. But again, there's a whole keep in mind that I'm in some ways doing this injustice because there are a bunch of proteins at each level which interact with these. There's many binding partners that are associated with these different complexes, but the CDKs and cyclins is where we can realistically kind of focus and not go overboard. Yeah.
Can you, on the last slide, can you just repeat that from the last point kind of
The last point? We're going to go back into the last point later. If I forget, remind me. So, for each phase, G1 phase, okay, make sure you understand G1 phase. So, we talked about a couple of proteins and things, but make sure you know the importance of each phase. So, G1 phase, cell growth, repair. The cell checks the metabolism, the metabolic activity. It regulates its cell growth. It's involved in cell growth, that stage. It checks the DNA. So all these things, before the cell decides whether to start replicating the DNA, things need to be checked. And then when they're happy, then you can start generating, doing DNA replication. This is based on intracellular signals that are basically going to talk to that G1 stage, regulating primarily the CDK, the cyclins, and then a couple of other proteins. And one of the proteins that we're going to talk about in a minute is P53. Before we talk about P53, it's the same thing for mitosis. So for mitosis, the M phase, make sure that you understand the fact that MCDKs are important and the fact that these cyclins and MCDKs are switching off rapidly, fast and slow to regulate the mitosis process. Okay, and we'll talk about this a lot more next in 2 classes, 2 lectures. So when DNA is damaged, and this is something that a lot of researchers have been involved in a lot of labs for cancer, because this is where a lot of the cancer cells arise from controlled proliferation, cells that divide that have got problems with the DNA. When you've produced your 2 cells, and then G1, you need to check those cells. You need to make sure the DNA is good and everything else. If there is any evidence of DNA damage, you can activate a cascade of events. You need to know this for the exam. I will ask you questions on this, but this is one of the most heavily studied proteins on the planet. P53. There's probably 1,000 labs, more than 1,000 labs in the world that study P53, because it has so many effects on the cell, and it's associated with so many disorders. So if you get some kind of DNA damage, you will first activate a series of PKs, protein kinases, and one of them will actually phosphorylate this key protein, P53. If P53 is activated, it can activate lots of things, and most of them we're not going to talk about today. But what we will talk about is the fact that when P53 is activated based on its phosphorylation state changing, it will then activate and bind to a particular DNA region of what will be involved in transcription of the P21 gene. So this P53 will bind to the DNA that's where that P21 gene is. If it binds to some sequence upstream of that P21 gene, it will cause transcription of the P21 mRNA. You'll get the P21 protein, and what it does is actually inhibit CDKs. So once this green is activated, this all makes sense, right? Think about it. G1 to S CDKs. For me to go from G1 into S, I need to trigger certain CDKs that are G1S dependent, that they only switch on to push the cell from G1 to S. If there is evidence of DNA damage, we need to stop that. And the way we stop it is by activating P53 pathways. And that knock-on effect produces a protein that comes along and binds to this cyclin CDK complex and stops it from doing what it should be doing. And that will hold the cell in G1. Does that make sense to everybody? P53 to P21, produce your inhibitory protein that will take the complex, stop it from doing anything. You will hold, you will stop at the G1 phase, and you will not move to S phase. Okay? If the damage can be repaired, and we have a series of molecular, you might do this. In molecular, we might do this. You might do this if you do molecular biology. You'll do DNA replication, of course, but you'll do P53. You'll do all them types of mechanisms and DNA repair. Really complex pathway, but absolutely essential for making sure DNA is good. Okay. P53. As far as P53, I mentioned about what it does in this pathway for G1, but make sure you understand in terms of this is just an example of the apoptotic mechanism that can be associated, because P53 will try and help in fixing the DNA, or will trigger the pathway which will stop the cell moving into S phase until it's time to do so. If the cell can't move through for too long, and if the DNA is unrepairable, or if there's some significant problem, what we don't want is that cell to say, hell with it, we're going to send it through anyway. Okay? That's what happens when you have mutations. In this case, we don't want that to happen. If we do say to the cell, you cannot go to S phase, we cannot replicate your DNA, then it will activate, and for you guys, you just need to know for the exam, you will activate the apoptotic pathway, and you will cause that cell to get killed. Okay? You have a question?
Yes. No, I don't think.
You don't? What?
Wait, so if P53 has a mutation in it, then it gets...
Well, that's another thing we haven't talked about. If if P53 has mutations in it, then all types of bad things can happen. But one of the things that can happen as far as cell cycle, if P53 mutated, then you'll get DNA damage that arises, and it won't necessarily be stopped there. And instead of me stopping it when the DNA is damaged, I can send the cell through to S phase, even with damaged DNA. So that's one of like 50 different things P53 does. Okay, there's lots of things. Yeah.
So P53 is located close to the cell cycle? In case of something happens, it can then go to the
so
DNA?
P53 can be transcribed and translated quickly as well. It's a protein, right?
Okay.
But it's also in the vicinity of regulating G1. So when the DNA is being checked, and if there's some DNA repair mechanism that says, right, okay, there's damage here, we need to, before we even think about repairing it, we need to stop the cycle. P53 will be activated, and then it will be like lots of proteins that are transcription factors or have a DNA binding domain. They will bind to a gene. In this case, P53 binds to P21 gene, but by doing that, it regulates transcription of P21. By producing P21, you produce an inhibitory protein that will then go to the cyclin CDK complex and stop it. And until the DNA is repaired, hopefully, nothing else is going to happen. But if you have mutations in P53, you lose that. And so then you start to get S phase happening in cells that shouldn't be going through S phase. Yeah.
What's checking the DNA that causes the P53 mutation?
Oh, so we're not going to talk about that. That's a whole mess. I would love to talk about that. There's like 50 proteins involved in regulating DNA. So you'll do DNA replication, you'll do DNA repair. It's a long process. So you can come and see me, we can talk about it. Just time it. You have a question?
Yeah, it's similar to Austin's, but so the activation of the P21 gene, does it only cause the inhibition, or does it also activate the DNA repair process?
No. What this process is primarily for is to stop active SCDK from becoming to do what it wants to do, which is to SCDK is involved in the pre-initiation complex for replication. You don't want that to happen, so you need to stop SCDK, and that's where the inhibitory protein is. So just think about it as a stage thing for now. It's just stopping the stage from moving forward. P53 is a big deal. Okay. So as far as S phase, S phase is very complicated in terms of lots of things that happen, and the repair mechanisms are very complicated. Lots of proteins involved. And but at least with me, when I did an advanced cell 10 years ago, it was 4 or 5 lectures just on DNA repair. So it's a lot of proteins playing a role. But in terms of the S phase replication, the DNA replication, there is a series of proteins that are involved in the initiation part, just like transcription I mentioned before. So for transcription to happen, you have to have all these guys coming in doing things. And it's the same thing for S phase, for DNA replication. So for the mechanism, if there's no mutations or if there's no obvious DNA problems, the DNA will be primed to go through and presented to go through the replication pathway. Okay? In other words, start to get ready to go into S phase. And what happens is at the late G1, you will get a protein complex. So DNA-made replication. This basically is an iteration of the diagram. So when you're reading this, look at this diagram. It obviously looks a lot better. But use the text that's here because I put it in to help you explain the mechanism. The DNA is made replication ready. What that means is there are specific sites on the DNA called origin of replication sites. They're initiation sites where a bunch of proteins are going to converge onto that place. Imagine it's similar, if you like, to a TADA box. There's specific sequences in there where a bunch of proteins involved in initiating DNA replication need to come in. So this origin of replication complex needs to sit here, and the protein CDC6 is involved in this initial docking, if you like. Then what happens is there's a protein called helicase that comes in, and this helicase actually binds and removes this CDC6 from the complex. Now you've got once you've got this origin ORC complex here, and you've got the helicase, this whole thing is called the pre-RC, pre-replicated complex. So now if you're at this point, the DNA looks like it's nearly ready to be replicated. Because you've got a site which is the target where this is going to be. You've got this potentially scaffold, this fully functional pre-replicated complex, which is a helicase and the origin replication protein complex all together. Okay. Then helicase, once it's activated, will then also bring in, or the protein that will now come in, the protein complex that will come in, will be SCDK and recycling. This is where the SCDK and the cycling comes in. So the SCDK and cycling comes in, and actually at this point here is involved in promoting this process. And what happens is, using this helicase and using a polymerase, and now for this we're going to make DNA upon DNA, right? So you have a DNA polymerase. What did we have before? We had an RNA polymerase, because you produce an RNA from a DNA strand. In this case, you have a DNA, which is just replicating DNA. So you then activate a DNA polymerase. And what happens in this whole process, where the helicases and where this DNA polymerase is, the DNA polymerase is going to produce a new strand of DNA. And we're not going to go too detailed with this, because like I say, you're going to do this, hopefully, if you do molecular biology. But what you produce here is it allows this DNA to open up. And in this place where you actually initiate this replication, it's called a replication fork. It's where the DNA runs and then suddenly it opens up like this. Okay? So, CDC6 is there. CDC6 will come off because this ORC helicase will come in. You get this pre-replicated complex. Once that's there, then SCDK and the cycling will play a role. And we're not going to talk about the details of that. Then, once this helicase is basically come in, you'll see the replication complex is then, you'll have a recruitment of DNA polymerase. The DNA polymerase will start to produce the new strand of DNA. And you will get DNA upon DNA. And so there's lots of these across the DNA, not just one of them, right? Just like translational ribosomal sites, you have lots of these set up, these sites or docking sites for all these complexes all the way along the DNA, so you can replicate the DNA quickly. If you had one, it would take years to do. Okay? Somebody have their hand up, I think? Yeah.
So CDC6,
CDC6.
the purpose of that is just to help DNA helicase bind on.
So SCDK and the cycling here is involved in this place here in terms of promoting this fact.
CDC6.
Oh, CDC6 is up here.
Yeah.
Right. So CDC6, once that's there, nothing's going to happen. And when that's removed, because when it's removed is when the DNA helicase comes in, and that dissociates the CDC6. Yeah.
I'm a little confused on that too. So
Sure.
so you have the DNA strand, and then the ORC binds first, and then CDC6.
ORC is there, and CDC6 keeps hold of it while it's there, and then when the helicase comes in, it will dissociate the CDC6.
Oh.
And then helicase can come in and sit right in front of, upstream of this pre-replication complex.
So CDC6 is there just to kind of inhibit.
Yeah, it's there as a inhibitor of the process.
Okay.
And then once you come in with the helicase, you can basically alleviate that and then start to activate recruiting various proteins. The replication complex, the helicase, the SCDK proteins, the DNA polymerase, and you start to produce the DNA within that S phase. And so you duplicate all the DNA. As far as this exam, because this again, it's all about knowing the cycle. For this exam, just make sure you know these key proteins within this one diagram. That's what I care about as far as the S phase. Okay. Again, appreciate that this is it's a lot more detailed than this. But in terms of the directionality and who's playing a role, make sure you know these key proteins that are involved in promoting, allowing the S phase to happen. So when all these are in place, then you can start to drive S phase. Just going to move forward. Then you can start to drive S phase. And once you drive S phase, that means you've got the DNA ready. You've got it in a position where it can start to replicate, because you've got the pre-replication complex. You've got all the proteins on there, and then the DNA polymerase will start to produce DNA. Will start to replicate the DNA. And so that's critical for being able to do the S phase. So last couple of things. We have a couple of minutes. So in terms of two key proteins, we talked about CDC25. Make sure you understand the importance of CDC25. We've already talked about it, but make sure you do. As far as regulating MCDK cycling, because that's the one that's involved in allowing M-cycling MCDK to go and push the space into M phase. Okay, so make sure you have that down. And make sure you have the mechanism down in terms of phosphorylation state. Dephosphorylation using CDC25 is involved in activating it. The actual phosphorylation through WEE1 keeps it inhibited. And it's that flip-flop between them two states that allows mitosis to happen. Okay? I'm not going to say any more, except, so on Friday we're going to do, just the way I got it right now, we're going to do muscle contraction. On Wednesday we're going to do mitosis. On Friday we're going to do cancer, and then unless I'm mistaken, the following week will be a review session, and then it'll be the final. Okay? Questions about this? Key proteins in each place, where they're involved in which stages of the cell cycle. That's what I care about for this. This is kind of setting up everything to then go into mitosis. Okay? So when you're reading through this lecture, use that strategy. Exam page in the email,